Hello dears, today I am with you to discuss another very important topic in physiology, which are the blood groups. In the membrane of human red cells, RBCs, there are certain molecules identified which has the ability to act as an antigen. So what an antigen is? An antigen is a substance or a molecule that can evoke an immune response in a person, just like a virus. It can evoke an immune response. So just like a bacteria, it can evoke an immune response in a person. So such molecules, certain such molecules are identified in the membrane of RBCs, okay? And depending upon the antigen, different persons, each person is unique, each person is different, and depending upon the antigen, the person, the individuals can be classified. So blood group is based on such antigens present in the human red cells. The antigens are called agglutinogens, and there are certain antibodies in the plasma. So antigen, remember, antigen is in the cell membrane and antibodies are in the plasma. These antibodies are called agglutinins. So depending upon the antigens, depending upon different antigens present in the surface of the cell, red cell membrane, there are 30 different blood group systems identified. Okay, there are 30 different blood group systems identified. But most of these antigens are inactive in our temperature. It could be active in cold temperatures only. So most of the antigens are inactive. So only ABO system and RH group systems are most important in case of blood transfusion. So in case of blood transfusion, you are receiving blood from outside, not from your body. It's not prepared from your body. You're receiving blood from an outside source. So in order to stop immune response, you have to identify the blood groups. You have to identify the antigen, and you should know that whether this blood transfusion will evoke an immune response in the person. So regarding transfusion, ABO and RH group antigens, AB antigens and RH antigen is the most important, which are able to arise an immune response in the recipient. So these blood groups are most important. But other major blood groups, you should know the names. They are MNS, Lewis blood group, Duffy, Kell, P, and Lutheran systems. So what are the uses of blood groups? First and most important use we have discussed already, to check the compatibility for blood transfusion. So during blood transfusion, the donor will be donating his blood to the recipient, and if this blood contains an antigen which can evoke an immune response in the recipient's body, then this blood transfusion will give hazardous effect, even life-threatening effects. So it's very important to check the compatibility using blood groups, and that is why your license is having an option for blood group. Then the blood group antigens have provided the scientific basis for understanding hemolytic disease of the newborn. So this hemolytic disease of the newborn occurs due to RH incompatibility. We have studied ABO and RH blood groups are important. So this hemolytic disease is due to RH incompatibility. This RH antibody can cross the placenta and cause hazardous effect to the baby. So it's very important to know the RH incompatibility also. And next use of blood group is to determine the susceptibility to infections by certain parasites, bacteria, and viruses. So there are certain hypotheses regarding different organisms, like AB and B blood groups are more prone to salmonella infections, E. coli infections. Then Vibrio cholera have more susceptibility to infect O blood groups. Like that, there are many hypotheses. And then red cell antigens are also associated with clinical disorders. So, like certain blood groups are prone to certain diseases. For example, AB blood groups are more prone to memory loss. Then, blood group A is more prone to cancer, especially stomach cancer. Then, ABO genes, you will study about the secretor group. The secretor groups are more prone to coronary disease, heart attack, stroke, etc. So, such that, like that, many hypotheses are there. And then, next is disputed paternities. So, disputed paternities means if you want to identify the father, these blood groups can provide a hint regarding if this particular person is father or not, but it is not very confirmatory. We will see how it is determined. We will see that in the coming classes, coming slides. And then certain studies say that blood group influences personalities also. Or certain personalities can be common to certain blood groups. Like blood group A, persons will be stubborn, they will be tense or reserved. Then blood group B, persons are more creative, they will be strong but unforgiving. Then blood group AB, they are very controlled persons, cool persons, but they may be forgetful. And O blood group, they are confident, workaholic, but they are unpredictable. Like that, certain hypotheses are there regarding the personality types and blood groups. So blood group has an area used in there also, determining the personality. So ABO system, as we already discussed, is the most important among the blood groups. It was discovered by Karl Landsteiner in 1900s, and for that he received a Nobel Prize in 1930 in physiology or medicine. Then, why ABO system is important? Because of the naturally occurring antibodies A and B in plasma. So, A and B are naturally occurring antibodies in the plasma, while RH is not. There is no naturally occurring antibody for RH. Important. There is no naturally occurring antibody for RH. You develop antibody only after exposure. But apart from that, A and B antibodies are naturally occurring inside the plasma. We will study about who persons have which person have which antibody. We will study that. But because of the naturally occurring antibodies, these ABO blood group, incompatible ABO blood groups, instantly leads to serious consequences in blood transfusion. So it's very important to study the ABO blood group and the compatibility. So in the ABO system, there are two antigens identified, antigen A and antigen B. A antigen has again two subtypes, A and A1. And where is this present? These antigens are present on the membrane of red cells. They are also found in many other tissues like salivary glands, testes, lungs, kidney and pancreas. So they are secreted in minute quantity in saliva, pancreatic secretion and seminal fluid. See, RH antigen is only present in RBC membrane, whereas A and B antigens are present in some other tissues also. Then, in case of blood, so we studied that there are only two antigens, antigen A and antigen B. There is another antigen called H antigen. We will discuss about this H antigen later. When we discuss the formation of this A and B antigen, we'll discuss the H antigen. But now you just know that there is an antigen called H antigen, and O blood groups secrete this H antigen. And then, when does this ABO agglutinogen appear in our bloodstream? So, it is present since fetal life. They first appear at 6th week of intrauterine life. So, by birth, you have this antigen in your RBC membrane, and it is only 1/5 of the concentration of adult life. And in course of time, when you reach your 15 to 16 years of age, this quantity of antigen will peak. And that is why you should have a minimum of 15 to 17 years for being a donor. Okay. So why? You need 15 to 17 years because of the presence of this antigen. This will reach a maximum at 15 to 17 years of age. The naming of blood group is based on the antigen present in the blood group. So blood group A will be having A antigen, B will be having B antigen, AB group will be having A and B antigens, and O group will be having no antigens. And we have studied that A antigen has two subtypes, A and A1. So blood group, there is blood group A1 and blood group A2. In the blood group A1, you have both subtypes present. That is blood group A antigen and A1 antigen. And in blood group A2, you are having only A antigen present. So in AB blood group, it will be subdivided into A1B and A2B blood groups. Next is distribution of antigens. So O is the most predominant blood group in the Indian population. Then comes a B, then A, and then AB. And in A blood groups, A1 is 75% and only A antigen present, it is 25% only. So there are 3 genes in the ABO blood group. These 3 genes are located on the 9th chromosome. We have studied that. Okay. And the first gene is H gene that secrete a basic precursor substance called H substance. So in presence of A gene, this H substance will be converted into A antigen. In presence of B gene, this H substance will be converted into B antigen. And if no A and B genes are present, that is in O blood group, this H substance will be the H antigen itself. So A blood group will be having A antigen and H antigen. B blood group will be having B antigen and it can have also H antigen. O blood group will be having H antigen. So these 3 blood groups are called secretor blood groups. And in case of Bombay blood group, OH, these individuals lack the H gene. So the basic precursor substance is secreted by the H gene, right? So this basic precursor substance is converted into A antigen, B antigen. So if there is no H gene present, then no H substance, no basic precursor substance. So no formation of A antigen, no formation of B antigen, even no formation of H antigen. So this blood group is a non-secretor blood group and it is called Bombay blood group. But their serum contains anti-A, anti-B and anti-H antibodies. So it's very important. The serum contains A, B and anti-H antibodies. So they should be transfused with only the Bombay blood group. So far we were discussing about the antigens present in the ABO system. There are 2 antigens, A and B. Antigen A is having 2 subtypes, A and A1. So likewise, there are 2 antibodies in ABO system. They are antibody for A antigen and antibody for B antigen. So these antibodies are called agglutinins. And just like antigen A having 2 subtypes, antibody for A antigen will also is having 2 subtypes. Then they are found in plasma. Then these blood group antibodies are naturally occurring antibodies. What does that naturally occurring antibodies means? Because when an antigen is exposed to the immune system, our immune system will be developing antibodies. So it is only after exposure to the antigen. But in case of this blood group antibodies, that is antibodies A and antibody B, they will be naturally occurring. They will be developing in our body naturally, and at the time of birth, there is no antibodies present in our body, and then they appear only in the second week of neonatal life. So during second week of our neonatal life, these antibodies will begin to appear in our plasma, and then they will reach peak level at about 10 years of life. Why these antibodies are forming in our blood plasma? The reason we don't know. The reason is predicted to be some intestinal bacteria or food containing such antigens, but we don't know the reason why these antibodies are naturally occurring or naturally forming inside our plasma. But they are naturally forming, and they are not present at birth. They appear only in the second week of neonatal life. Then what is the chemical nature of the antibody? They are IgM category. What is the importance of IgM, immunoglobulin M category? They cannot cross the placenta. And they are also called cold antibodies. Why? Because they efficiently work at low temperatures. So, anti-A antibody and anti-B antibody is of IgM category, which cannot cross the placenta, whereas Rh antibody is IgG antibody. It can cross placenta, and that is why erythroblastosis fetalis occurs due to Rh incompatibility. Next is Landsteiner's law. So before going in detail what Landsteiner's law is, let me remind you something. So what is an antigen? An antigen is a substance that evoke an immune response. And what is an immune response? During immune response, antibodies will be produced against the antigen. So an antigen and its antibody cannot coexist. Got the point? An antigen and its antibody cannot coexist together. If they coexist, that means there is an immune response. Okay, and Landsteiner's law has two limbs. The first limb say that if a particular antigen is present on the red cell membrane, the corresponding antibody will be absent in plasma. For example, if antigen A is present in the red cell membrane, then antibody for this antigen A will be absent in the plasma. That's very understood because the antigen and the antibody cannot coexist together. Conversely, if the agglutinogen is absent in the red cells, the corresponding agglutinin must be present in plasma. For example, if a person is having antigen A in his bloodstream, okay, then according to Landsteiner's law, first, there will be no antibody for this antigen A. And according to second law, a person is having A blood group. So, he is not having B antigen, right? So, this antibody B will be present in the plasma according to the second part of the law. So, this will hold good for ABO system, but it will not hold good for RH system. Why? Because RH antibody is not naturally occurring antibody. It will be produced only after the exposure to Rh antigen. So, Landsteiner's second part will not be applicable to the Rh system. It is applicable only to the ABO system. Based on Landsteiner's law, what will be the antigen and antibodies in the ABO system? So, blood group A. Why it is called A blood group? Because it has A antigen. That is why it is called A blood group. So, since it has A antigen, there will be no antibody A present in the plasma. It has no B antigen, so B antibody will be present in the plasma. In blood group B, it has B antigen, so there will be no B antibody present in the plasma. Instead, it will be having A antibody because it has no A antigen. Then blood group AB, because of the presence of both antigens, there will be no antibodies present in the plasma. And then in O blood group, because it is having no antigens, both antibody will be present in plasma. The Landsteiner's law can be applied to determine the blood group of a person. You have antibody A commercially available, antibody B commercially available. So you take a drop of blood, you add this antibody A to the drop of blood, and if agglutination happens, what does it imply? There is A antigen in it. So if there is A antigen in it, which is a blood group, it is blood group A. And then you take a drop of blood, you add antibody B to the drop of blood, and if agglutination happens, what does it imply? There is B antigen in it. So B antigen and B antibody reacted to form the agglutination. So what does it imply? It implies that it has also B antigen, and the blood group will be probably AB because it responded to both antibodies. So you can determine blood group, and process is called blood grouping. This we have already discussed. There are three genes. What are they? A gene, B gene, and the H gene. And if H gene is present and then A gene is present, the blood group will be possibly A. If H gene is present and B gene is present, blood group will be possibly B. And if H gene is only present and A and B genes are absent, the blood group will be possibly O. This we have already studied. So, the presence of these genes A or B or presence of antigen is a dominant characteristic. So, if the parents, father is blood group A and mother is blood group B, their possible genotype will be AO and BO. That means H gene and A gene is present. For mother, it is B gene and O gene present. And then the gametes will be what? It can be A, it can be O, it can be B, and it can be again O. So genotype can be four types. That offspring may be of AB blood group, the offspring may be of A blood group, offspring may be of B blood group, and offspring may be of O blood group. Hope you understood. Next is regarding the RH system. It is the second most important blood group system in transfusion medicine. So this was also identified by Landsteiner and Wiener, but in 1940 we studied that the other blood group ABO is identified in 1900. So this is first identified in rhesus monkeys, and that is why it is called RH system. And in the RH blood group system, there are 6 antigens identified: C, D, E, then small c, small d, small e, and immunologically the most active antigen is D antigen. So AB, ABO blood group, then C, D, E, RH blood group. Thus in RH system there are mainly 2 blood groups, which is whether the D antigen is present, which means it's RH positive, and if D antigen is absent, it is called RH negative group. So RH positive, RH negative group. 2 blood groups in RH group. We have seen that RH antibody is not a naturally occurring antibody. It will be developed only after an exposure. So if an RH negative person receives blood from an RH positive person, it requires time to build up the antibodies. RH negative person, at first when he receives blood from an RH positive person, his immunity will identify the antigen present in the RH positive blood, and it will start developing antibodies, and these antibodies will reach a significant titer value only after 3 to 4 months. You know that an RBC, lifespan of an RBC is only 120 days. So by the time the immune system reaches a significant titer, what will happen? This RBCs will be dead. So no immune response will take place. But if that same person receives an RH positive blood in the second time, what will happen is the memory cell starts synthesize antibodies immediately, and that will give rise to RH incompatible hazardous blood transfusion. So that is the importance of identifying RH blood group. Then this RH antibody is of IgG type, which can cross the placental barrier. So the same theory will be applicable there also. For example, an RH negative mother is carrying an RH positive child. At the time of delivery, at the time of childbirth, during placental separation, some amount of fetal blood gets mixed with the maternal blood. So what will happen? Maternal blood will be exposed to the RH antigen, and it will start developing antibodies. So when this mother became pregnant again with this with an Rh positive child, this Rh antibody can cross a placental barrier and it can affect the fetus. So that is a basis of formation of erythroblastosis fetalis or hemolytic disease of the newborn. And this Rh antibody is a warm antibody. We studied that AB antigen, AB antibodies are cold antibodies. Here Rh antibody is a warm antibody. So this we have discussed in the previous slide. It is erythroblastosis fetalis, and this disease occurs as a result of Rh incompatibility when an Rh negative mother carries an Rh positive fetus during pregnancy. We have discussed that at the time of placental separation, a small amount of fetal and maternal blood get mixed. Then maternal blood is exposed to the antigen. It will start making antibodies, and when this mother is again carrying an Rh a positive child in the next pregnancy, what will happen is the memory cell starts synthesizing Rh antibodies very rapidly, and this antibodies can cross the placenta and affect the fetus, and it will produce a disease, erythroblastosis fetalis or hemolytic disease of newborn. What will be the clinical features of erythroblastosis fetalis? Due to the antigen-antibody interaction, the red cells will be hemolyzed or destructed. And due to increased destruction of red blood cells, what will happen? There occurs anemia. And due to the destruction of red blood cells, bilirubin in the blood will increase. How? Because the bilirubin is formed by the destruction of RBCs. You know that, right? So that will cause hemolytic jaundice, means increase in bilirubin due to increase in destruction of RBCs. Then generalized edema. Because of anemia, body will start producing more and more amount of what RBCs, and in order to produce RBC you require protein, more protein will be used up, and so the protein store will be depleted, and that will give rise to less amount of plasma proteins. Less amount of plasma proteins means edema. So this is called high fetalis, means there is anemia and also hypoproteinemia. We have discussed that due to over destruction of RBCs, hyperbilirubinemia occurs, and if the bilirubin is deposited in the basal ganglia, it will give rise to neurological syndrome which is called kernicterus. But in case of adults, you know, the blood-brain barrier is very much developed. What is blood-brain barrier? There is a barrier existing in the central nervous system, and this barrier will allow only some things to pass through this barrier to reach the neurons. But in case of children, in case of newborns, this barrier is not very well developed. So this bilirubin will cross the blood-brain barrier and it will reach the basal ganglia, get deposited there, and that will give rise to the disease kernicterus. So after delivery, hemopoiesis takes place in bone marrow, but because of the severe anemia, there occurs extramedullary hemopoiesis, means other structures like liver start producing RBC, and so erythroblast will be released into the blood. What are erythroblast? They are nucleated red cells. Actually, our RBCs are non-nucleated, but erythroblast will be immature RBCs will be released into the bloodstream, and they can be detected in the peripheral smear. So it is due to extramedullary hemopoiesis. So this is how erythroblastosis fetalis is prevented. That means immediately after the delivery, you give some single dose of anti-Rh antibodies to destroy the antibodies formed in the maternal blood. Okay, so that's a prevention. Then next slides will be about different blood groups other than this ABO and Rh system. You just read the blood group, just study the names of the blood group, like L blood group, Lewis blood group, like that. You study the names. Do write notes. This is a 5-mark question, frequently asked. So do write notes.